Hello, and welcome to the second lecture in my fourth lecture series. The lectures in this series concern some basic aspects of pharmacology. In the last one, I talked about some features of pharmacokinetics, and as you can see in this lecture, I'm going to focus on pharmacodynamics. Now, of course, later in the semester, when I do more lectures, I'll be focusing on specific drugs, and for each of those drugs, I'll try and hit some important points about pharmacokinetics and pharmacodynamics. But my goal for these two lectures, as for this lecture series, is just to give you some basic information that I think will serve as a foundation for some of the stuff that we'll talk about a little bit later. So for today's lecture, I want to introduce the word psychopharmacology, which you may be familiar with already, or which you could probably define just based on the word itself. I want to talk a little bit about what psychopharmacology is, and then I'm going to focus a little bit on one part of pharmacodynamics, and that is tolerance. It's a, a phenomenon, or really a set of phenomena, which I've mentioned before, which you're probably somewhat familiar with already, but which can be kind of complicated and interesting and really deserving of their of some special attention. So I want to talk a little bit about tolerance, some of the mechanisms for tolerance, and then we'll kind of wrap up this lecture and wrap up this lecture series, and then be able to move on to talk about, in future lectures, some specific drugs. Okay, so that word psychopharmacology, I introduced it just a second ago. Recall that pharmacology is just broadly defined as the study of drug dosing and drug effects. So you could probably guess just from the word that psychopharmacology is the study of drug dosing and psychological effects. Really, all the drugs that we're going to consider in this class are drugs which are psychoactive, which have effects on various aspects of the mind, of the brain's processes, that have effects on how we think, how we feel, and how we behave. So really, we're mostly talking about psychopharmacology, but I held off from introducing that word last time because I felt like I was already throwing a lot of fairly complicated terminology at you. Recall last time I showed this graphic just to kind of put forward the idea that pharmacokinetics and pharmacodynamics are two aspects of pharmacology. For our purposes, we can just say that you could consider within pharmacology, psychopharmacology being any of the study of drugs which have psychological effects, and we can consider the psychopharmacokinetics and the psychopharmacodynamics of various drugs if we want to use a lot of very complicated words. So again, psychopharmacology, the study of how drugs affect things that the brain does, things that that we are interested in psychology, things like thoughts, feelings, and behavior. And often in psychopharmacology, there's a real interest in those behavioral effects of drug use. Not that the other stuff isn't important, but a lot of research, especially a lot of research using non-human animals, of course focuses on behavior because it's most easy to observe and to measure. Psychopharmacology is also, in many ways, the study of the various factors or variables that modify the effects that drugs have on thoughts, feelings, and behaviors. And there are a great many different variables, biological, psychological, and social or environmental variables, which can modify the effects that drugs have on people, that can change the way the feeling of alcohol intoxication is from one person to another, or from one person at one time to that same person at another time. And it would be very complicated to talk about all these things right now in this lecture, so I'm going to really put off a lot of that discussion till later in the semester when I can focus on some of those factors for specific drugs. What I do want to focus on today is that phenomenon of tolerance, which I mentioned already. Tolerance is one of those factors or one of those variables which changes the way drugs are experienced, changes the effects that drugs have on people in terms of their thoughts, their feelings, and behavior. And like I said just a slide or two ago, I think tolerance really deserves a lot of attention and really can be kind of complicated and interesting to think about. So it's well worth, doing just that in this lecture. tolerance is an interesting phenomenon to talk about, in part because we almost all of us have some experience of it already. Those of you who consume alcohol, those of you who use nicotine products probably are familiar with tolerance. Almost all of us use caffeinated products of one sort or another, whether they're coffee, tea, sports beverages, etc. So you probably know a little bit about tolerance for those drugs, say caffeine in particular, even if you haven't really thought too much about it yet. So simply defined, tolerance is a phenomenon that occurs following repeated use of a drug. So if we continue using a drug at a same or a fixed dose over time, we typically find, for most drugs, not all, but for most drugs, that the effect of each dose decreases. So a drug like caffeine, which is found in an espresso, sort of cup of coffee, will have a given effect or a given set of effects on someone, maybe making them feel more alert, maybe making them feel a bit jittery, increasing their heart rate, etc. Those effects will tend to decrease in their amplitude or decrease in their size or potency over time if that person keeps on using that same dose. He or she has the same size cup of coffee every day. The effect after a certain number of days of dosing in this fashion will be less than those effects on the first day when that dose was first introduced. And we can observe this in humans, we can observe this in non-human animals very easily for most drugs that we're interested in. What often goes along with tolerance, though, is, at least in humans or in animals who have access to varying quantities of drugs, who can choose to use more drugs if they want, is that people will tend to increase their dose of a drug as tolerance occurs, so as to offset that tolerance, so as to offset the declining potency of the drug with repeated use. So again, to be simplistic about it, you've probably had this experience before. If you drink, let's say, coffee in the morning, you may find that when you first started drinking coffee in the morning, 1 cup of coffee or a small cup of coffee was enough to make you feel kind of alert and ready to go. But you probably found over time that same dose, that same size cup of coffee didn't really give you as much of a boost as it used to. And you may have adopted the practice of having 2 cups of coffee in the morning, or having a large cup of coffee, or having a stronger cup of coffee brewed with more coffee beans, or with additional shots of espresso or whatever else added into it. If you did this, whether you realized it or not, what you were doing is trying to offset the tolerance that your body was developing to some of the effects associated with the drug caffeine. Now, that sounds simple enough, and in some ways, of course, it is simple enough, but there are a couple interesting points that follow from it. One is that there actually are different types of tolerance that can occur for different drugs, and they're worth talking about individually. And there's also a question that lurks behind this, which is, why is it that we notice tolerance for most drugs, or at least most of the drugs that we're interested in? Why is it the case that the body experiences a decreasing set of effects for a given dose of drug if that dose is given repeatedly over time? So we'll try and explore some of the types of tolerance in the next few slides, and also some of the mechanisms that lie behind these different types of tolerance, so that hopefully this simple idea can be appreciated in its complexity, and you can maybe get a bit more of an understanding of how it all works. So the first type of tolerance to talk about is what's called dispositional tolerance, or it's often called dispositional tolerance. And again, the idea of all forms of tolerance is that with repeated use of a drug, the effect of each dose decreases. So that's the basic idea of tolerance. When we're talking about dispositional tolerance, we're talking about tolerance that occurs because of changes in metabolism or biotransformation of the drug in the body. So recall last time in my last lecture when I talked about pharmacokinetics, I talked about this idea that once a drug is introduced into the body, and maybe it begins to have effects because it's interacting with various parts of the nervous system, at the same time the body is working fairly hard to remove that drug. Different areas of the body are metabolizing the drug. For humans, and actually for most animals, the liver is a major place where drugs are metabolized, so the body is getting rid of the drug as quickly as it can so as to get rid of those effects. Your body usually, or generally speaking, fights against being intoxicated by almost any drug that you introduce to it. And that rate of metabolism, that rate of biotransformation can change, at least for some drugs. And so in this case, we have the drug being reduced or we have the effects of the drug being reduced because metabolism is increasing for the drug, and the drug, the body is more efficient at getting rid of the drug. This is sometimes called pharmacokinetic tolerance for a reason that hopefully seems kind of obvious given what I've just said. It's about the pharmacokinetics of the drug. As the pharmacokinetics of the drug, or at least the portion of pharmacokinetics that concerns elimination of the drug gets more efficient, there's less drug available for the nervous system. So the nervous system just isn't getting as much of that alcohol or as much of that cocaine or as much of that whatever as it used to after you've repeatedly used a drug, because your liver and other parts of your body are just much better at getting rid of the drug before it can really start to have its effects. Now, if you've ever had a checkup at the doctor, and of course I'm sure almost all of you have, you may have had a blood test done. And blood tests can be done for all sorts of things, of course, like blood sugar, blood lipids, etc., etc. Sometimes blood tests will include a measurement of various enzymes to indicate your liver activity. I'm not sure if all people have this done. Maybe it's just my age. At this point, when I go to the doctor and get a checkup done, I think this has become kind of a standard thing that is done. The measurement of those enzymes in your blood is a way of kind of estimating how active your liver is. And if you were someone who'd abused drugs, especially abused alcohol, for instance, you can over time damage your liver, making it less efficient. So you could, for a time, have some increased tolerance to, let's say, alcohol, because your liver is becoming more efficient at getting rid of the drug. But then after some point of long-term abuse or sort of very heavy abuse, you can actually damage your liver, and that dispositional tolerance can go away quite dangerously for your body. Another maybe more common example of that is that over time, people, as they age and get older, their livers just get less efficient. Your grandma, your great aunt, your grandpa, by virtue of their age, have less efficient organs in their body. At least many of their organs are, including their livers. So their liver is less good at getting rid of drugs than it was when those same people were younger. They tend to have lower dispositional tolerance for drugs than do younger folks. So dosing medications for elderly people can be quite tricky, because you want to give them enough to get an active dose, but not so much that they get a toxic or even lethal dose. That's all sort of having to do with dispositional tolerance, the effect of tolerance that arises from changes in metabolism for a particular drug. Okay, that's one form of tolerance. Another form of tolerance is called functional tolerance. And so again, the basic idea of functional tolerance is that with repeated use of a drug, the effect of a given dose decreases over time. That's basic. Functional tolerance refers to the idea of when this effect occurs, when this tolerance occurs, because the nervous system itself, especially the central nervous system, is becoming less sensitive to the drug. So dispositional tolerance refers to how quickly the body can get rid of the drug before it can even really start to affect the nervous system. Functional tolerance refers to the idea that even once the drug does come into contact with the nervous system, especially the central nervous system, because we're talking about psychopharmacology, the nervous system itself will get less sensitive to many of the drugs that we're interested in. And so any given dose, any fixed level of the drug, will tend to have a smaller and smaller effect over time. Now this is sometimes called pharmacodynamic tolerance, because it really emphasizes changes in the nervous system, how the nervous system responds to the drug that underlie this form of tolerance. So functional tolerance, pharmacodynamic tolerance go together. Previously, of course, I introduced dispositional tolerance. That is synonymous with pharmacokinetic tolerance. Okay. So we sometimes, if we want to be a bit fussy or a bit complicated about it, we can distinguish between acute and more long-term or protracted protracted functional tolerance. Just to be a little bit clearer about that, acute functional tolerance is it occurs when, or is observed when within one session of drug use, the effect of the drug changes over time, or the sensitivity of the nervous system changes over time. To break from showing all these pictures of coffee, I'll show a picture of another pretty common drug, that's alcohol. Of course, in this case, in an example, in a picture of beer. Alcohol is a drug where you can sometimes observe fairly good acute functional tolerance to the effects of the drug. So, for instance, alcohol tends to impair short-term memory at the doses that most people consume alcohol at. Those effects are not particularly strong, nor are they particularly long-lasting, but they can be observed fairly reliably. So if I was to give you a task that measured your short-term memory, for instance, asking you to memorize sets of letters or sets of numbers or symbols or things like that, and then I would get you to give them back to me after a short period of time, thus testing your short-term memory. And while you were doing this, I gave you a dose of alcohol in a beverage. I would observe a greater impairment in your short-term memory from the alcohol as your blood alcohol level was rising than when it was falling. So if I picked an arbitrary point in your blood alcohol curve, like when your blood alcohol level had risen to 0.06 or 0.08, and I measured your level of impairment as your blood alcohol was going up, essentially as your body was getting more drunk, and then I measured your short-term memory at the same point, at 0.08 or 0.06, when your blood alcohol was falling as you were sobering up, I'd notice a greater impairment on that ascending limb earlier on in your drinking episode than on your descending limb of intoxication later on in your drinking episode. So you can see here, this is just an attempt to depict that. I can see, or you can see here in this graph, we've got 4 different people, and we are getting them intoxicated to different levels of intoxication, and you can see that there's a line that sort of divides the y-axis at the midpoint at 0.5 gram. I'm guessing 0.5 grams per 100 milliliter estimated blood alcohol content. The idea is if you tried to measure, let's say, someone's short-term memory as they were getting more drunk, as the curve was rising, you'd see a greater impairment than as the curve was falling. If you did, that would be an example of acute functional tolerance. The brain, by the time you're sobering up, even though you're still drunk, your brain, your nervous tissue has become less sensitive to alcohol. So the presence of that alcohol isn't causing as much of an impairment in one of the aspects of intoxication, impairment in short-term memory. Okay. Another example of this can be observed when we look at the subjective pleasurable effects of cocaine. So if we look at one session of using cocaine, where someone might use several different doses across a given session, like within a period of half an hour or an hour of an evening when they're using cocaine, typically what happens is cocaine users will report the greatest amount of pleasure associated with that first use of cocaine, that first line or bump that they inhale, and much less with later uses of cocaine. So their nervous system is becoming is becoming less sensitive to the effects of cocaine, and thus they're getting less of a boost or less pleasure from each subsequent dose that they're using within one session of cocaine use. Now this has some interesting effects for cocaine users. You could imagine for the first use, the first line that the person snorts, they have a strong feeling of pleasure, but it quickly wears off because cocaine is a fairly short-lived high. So they go ahead and use a second line of cocaine, they sniff a second line of cocaine, and this doesn't produce as much pleasure as the previous one, so maybe they do a little bit more in an attempt to kind of get a bit more of that pleasure back, but the effects quickly wear off, because again, that's what happens. And then the third use of cocaine doesn't produce as much pleasure as the previous one, so maybe they try and use a little bit extra, and the effects quickly wear off. And you can imagine for someone behaving in this way, using cocaine in this way, they might quickly go through a lot of cocaine in an attempt to recreate the full pleasure that they got from that first use. But they're unlikely to be able to do so because they're fighting against acute functional tolerance. And they may have to use quite a bit more cocaine to get back to that initial pleasure. They may not even be able to get back to that initial pleasure. And it is just something that we tend to see with cocaine users. They tend to use cocaine pretty much until their supply is gone. So people, I'm sure there are people who are able to buy cocaine and keep it, and save it for later. But what often happens with cocaine users is they'll begin using cocaine intending to use just a little bit, and they'll end up using quite a bit more than they'd intended within one session of use. Of course, this can be dangerous because along with the pleasurable effects of using the drug, there are some dangerous effects of using the drug, including a very sharp increase in heart rate, which can lead to a variety of cardiovascular problems in the body, and can of course even be fatal. So that's functional tolerance, more specifically acute functional tolerance. We can make some further distinctions on the timeline of functional tolerance, but that's about as far as I'm going to go for this one. Let's move on and talk about behavioral tolerance. Behavioral tolerance, again, is an attempt to explain why, if you use repeatedly a fixed dose of drug, over time the effect of that dose decreases. That's the same as all other types of tolerance that I've talked about. But here, the reason why the tolerance is occurring is because the user is learning to compensate for the effects of the drug. So he or she is learning to offset the effects of the drug behaviorally. This is sometimes called learned tolerance for the hopefully obvious reason that it involves a learning process on the part of the user where he or she gets better with each subsequent use of the drug at kind of offsetting or negating the effects of that drug. So a common example of this is acute behavioral tolerance for alcohol. As I mentioned in the previous lecture, a pretty common effect of alcohol intoxication is motor discoordination. So people stumble when they walk, they sway when they walk, they can even fall over, of course, and you may know this if it's ever happened to you, if you've drunk too much alcohol, or if you've seen people, you've been to a party or been out at a bar and seen someone who's had too much to drink and they're kind of swaying and stumbling around the room. What sometimes occurs, and what can often occur for people if they drink repeatedly, is that they learn to walk with a slightly lower center of gravity and a little bit more of a sway in their movements so as to not stumble as easily. Now, this doesn't entirely remove or entirely offset or counteract the impairment from alcohol, but it does compensate a little bit, and it's something that drinkers, at least repeated drinkers, can learn to do. I'll probably put a link on Blackboard, but you can certainly click around YouTube and find no shortage of funny or alarming videos of people stumbling around drunk, trying to get home from the bar or the party. If you just type in drunk walking or anything like that, you'll get more videos than you'll ever really need to see. I'll link to a couple of them if I can find ones that are funny but not too horrifying. I mean, I don't like seeing people stumble and hurt themselves. Hopefully you don't either. Again, the idea here is that the person is kind of learning to offset the effects of the drug. A yet further type of tolerance is cross-tolerance. Cross-tolerance can occur when regular use of one drug leads to tolerance of another drug. So if you're regularly using a drug like psilocybin-bearing mushrooms, magic mushrooms, or one of the types of magic mushrooms, whether you realize it or not, you may grow or develop a tolerance for the effects of LSD. That's a kind of a classic example of cross-tolerance. Or if you're a regular alcohol user, you can become tolerant to the effects of barbiturates, which are a class of depressant drugs or sedative drugs. Now, really, this is just functional tolerance, kind of like I mentioned earlier, but it's functional tolerance that can occur across two drugs, or cross tolerance for drugs, because, or when two drugs share a common biochemical mechanism. So becoming tolerant to one drug leads you to become tolerant to another drug. And I already gave this example, but I'll repeat it again. Psilocybin and LSD influence the nervous system in a very similar way. They influence some of the same neurotransmitter systems, although they are different molecules. As a result of this shared kind of mechanism, if you use one drug repeatedly, LSD for instance, you'll become tolerant to psilocybin or vice versa. And there are some other examples that can occur with cross-tolerance as well. Okay, so I've talked about I've given a lot of different types of tolerance. I've probably taken a concept that maybe seemed a little bit simple or obvious, tolerance, and maybe made it more complicated, hopefully not too complicated. An important idea to focus on here is that tolerance is a complicated phenomenon that for many people and for many drugs can involve different processes. All involve a decreased response to the drug, and all can lead users, at least human users or users who have access to a variable quantity of a drug, to use larger doses or more repeated doses so as to try and offset or negate the tolerance. And to be clear, it's not always the case that you can look at someone and say, this person has become tolerant to alcohol, this person has become tolerant to caffeine. It's only because of behavioral tolerance, or it's only because of functional tolerance. For any person, for any drug, it may be a mixture of these different types of tolerance which are collectively producing an effect such that this fixed dose of drug which the person is repeatedly using each time is giving him or giving her less and less of those expected effects. So I said before that one interesting thing about tolerance, of course, is this idea that there are multiple, facets or aspects of tolerance, different mechanisms, different types of tolerance. Another interesting question is, why do people become tolerant to drugs? How do these different aspects of tolerance work? I've already said there are different processes, different types of tolerance. It probably won't surprise you to know that there are different mechanisms which explain, at least in part, why these different processes or types of tolerance work the way they do. So in the next few slides, I'm going to try to highlight a few of the mechanisms which have been proposed and which have some support in terms of explaining how different types of tolerance take place, or how they work within a person or within a non-human animal for that matter. Now the first of these mechanisms is cell adaptation. Cell adaptation is a mechanism that's proposed to explain how functional tolerance occurs. Now recall that functional tolerance is tolerance that happens when the nervous system becomes less sensitive to a given drug at a given dose. And cell adaptation is thought to be a manifestation or an aspect of the body's attempt to maintain homeostasis. Homeostasis refers to a state of balance within the activities of different body systems. So we can think of simple examples like if your heart rate accelerates, your body will work generally to decelerate your heart. If your digestive activity increases, your body will work to kind of decrease activity. Your body is typically trying to maintain a kind of a set point or a set range of activity within different systems, whether it's your digestive system, your cardiovascular system, or various parts of your nervous system. Drugs, all drugs, or almost all the drugs that we're interested in, will disrupt that balance in one way or the other. And when that happens, the body works to restore that balance, to push back against that disruption. And as I've mentioned in previous lectures, this pushback is sometimes called opponent process or opponent processes. So if we think of cell adaptation at the level of the neuron, recall that neurons communicate to one another using neurotransmitters. So if you can see here the axon terminal of one neuron and the dendritic terminal of another neuron. If the first neuron is trying to communicate to the other, it will release neurotransmitters which will drift across the synaptic gap and will interact with receptors on the postsynaptic cell, on the dendrite of the postsynaptic cell. when drugs enter the nervous system, frequently they have their effects by disrupting the activity of neurotransmitter systems, causing, let's say, a massive increase in the release of a neurotransmitter, stopping the recycling of a neurotransmitter that might normally be occurring, or changing the rate of that recycling. Neurons adjust to this or adapt to this by changing the number of receptors that are present on their dendrites, or changing the sensitivity to those receptors, or by changing how quickly or how slowly they manufacture neurotransmitters. In one way or the other, trying to offset that disruption, or kind of balance out that disruption that the drug is causing. Now, one example of this is the way the nervous system responds to the presence of MDMA. MDMA, or ecstasy, or sometimes called Molly, is a drug which causes fairly large release of monoamines, including dopamine, norepinephrine, and serotonin. And the high that people experience when they're using MDMA, the feeling of kind of strong positive emotions, and a feeling of love and connection to other people, and to some extent distortions in the way the world around the user is perceived, those are the result of the disruption in different monoamine systems in the brain. Neurons in those different systems react to that disruption by downregulating themselves, by becoming less sensitive to their own neurotransmitters, and by producing less neurotransmitters, so that the kind of massive increase that the drug is pushing is made less effective, is blunted because of this decreased sensitivity in the nervous system. And interestingly, this cell adaptation is thought to explain why people will sometimes feel sort of depressed or dysphoric after they stop using MDMA. So if someone goes to a party or goes to an outdoor festival and takes some ecstasy or some Molly, while there, they may feel very pleasantly high and sort of happy and in love with the world around them. But as the drug wears off, and as they leave and go home, they may feel a bit depressed, in part because now those monoamine systems in their brain, systems which are responsible for regulating emotions, are now kind of tuned down because they've been fighting against that tuning up process caused by the drug. So there's kind of a pictographic representation of how this might work. We have a serotonin neuron. Let's imagine it's somewhere within the mesolimbic dopaminergic pathway. That pathway also includes serotonin neurons, not just dopamine neurons. Suffice it to say, it's somewhere within the reward circuitry of the brain. And you can see here that normally what happens is the axon terminal of one neuron will release serotonin, and it will drift across the synaptic gap and will bond, or at least temporarily bind, to receptors on the postsynaptic side, on the dendrites of other neurons. Normally what happens is there's kind of a molecular recycling process where after a serotonin molecule bonds to a receptor, it will drift back into the gap and will get kind of sucked up by a molecular mechanism in the axon of the neuron, and essentially gets recycled so it can be used again and again. This is the normal process which occurs over very short timelines, milliseconds of time, in countless different synapses all throughout your nervous system. Here's just one synapse for one pair of neurons to kind of illustrate what this might look like, sort of. Now, part of why ecstasy works is it blocks that reuptake process. It blocks that recycling process, which has, at least temporarily, the effect of increasing the amount of serotonin that is drifting around in the synapse of the between the two neurons. So temporarily there's more serotonin available to interact with receptors than there normally would be, and this is thought to be part of the reason why people have such strong experiences when they're on ecstasy. They feel, instead of normal happy mood, they feel incredibly happy mood. Instead of feeling somewhat happy to be with their friends, they feel just in love with their friends, like these are the best people ever there could be. And then instead of thinking that they're having just a nice time at the party, they feel like this is the best thing that could ever happen to them because of all that extra serotonin drifting around and activating any available receptor. And again, this probably accounts for some of those strong feelings that people have, and even to some extent the disruption in the experience of the perceptual world around them, kind of changes in the way the world is perceived. MDMA is sometimes grouped in with hallucinogens because it produces some mild distortions in the way the world is perceived. Anyway, the short-term effects of ecstasy can be quite pleasant. Of course, people report elevated mood, but in the kind of after effects of the drug can be fairly unpleasant. In the period of hours and days following ecstasy use, people sometimes report feelings of depression, feelings of irritability, feelings like the world is not very rewarding or very pleasant place to be. And the reason for this is thought to be that those systems in the brain, those monoamine systems, especially serotonin systems, which had been kind of overstimulated by the presence of MDMA because it was blocking the recycling process, adapt to that overstimulation by becoming less sensitive. Those little receptors on the postsynaptic side of the synaptic gap, on the dendrites of neurons, decrease in number and decrease in sensitivity such that the available serotonin, when it's now back to normal and its normal pattern of circulation, has less have fewer receptors to contact with, and because the recycling process has been blocked, there's less available serotonin to release anyway. So the person went from feeling normal mood to feeling very elevated mood while the drug was active to feeling kind of a below elevated mood, a kind of a depressed mood following the offset of the drug. So that's one attempt to explain tolerance, or especially a specific kind of tolerance, that is to say functional tolerance. Another type of tolerance, of course, is behavioral tolerance, and this is thought to occur by the process of learning, as I already said earlier. So when you're becoming behaviorally tolerant, or you're developing learned tolerance to a drug, you're learning to behave in ways that counteract some of the effects of the drug that you've taken. So again, this learning process is an explanation, or at least part of an explanation, for why there is such a thing as behavioral tolerance. The body is learning to compensate for a drug. So again, this has to do with homeostasis, your body seeking to maintain a balance, not letting any of its systems become overly activated or overly deactivated. When drugs disrupt the balance in your monoamine systems, or the balance in your heart rate, or the balance in your sensitivity to pain, or the balance in anything else, the body works fairly hard to restore that balance or counteract that disruption. So again, I've used this phrase before, I'm just kind of repeating it here. We sometimes call this an opponent process, or a set of opponent processes. The idea being that when drugs push the body in one direction, the body pushes in another direction to kind of offset it. I've also used this example before. When you drink coffee, caffeine starts to speed up your heart, and your body reacts to this by starting to slow down your heart, or counteracting it by slowing down your heart. With repeated use, over time, that compensatory reaction, that slowing down reaction, gets stronger and stronger, producing less and less, or leading to less and less of an increased heart rate associated with caffeine, something that looks like tolerance to caffeine, at least tolerance in terms of caffeine's cardiovascular effects. Again, I've used this image before in previous lectures, but I'm just repeating it. Let's imagine that the red line here is the experience that a drug has, or an effect that the drug gives a user on its initial use, whatever that effect may be, whether it's a change in heart rate, or a change in serotonin activity, or change in anything else. Initially, that effect is quite strong, but over time the body learns to balance it out with these opponent processes, whatever those may be, such that with later drug use, the effect of the drug is blunted or limited, illustrated here in this kind of solid blue line on the second figure. So I've called this a process, or I've identified this as a process for explaining behavioral tolerance, which is sometimes called learned tolerance, which might lead you to wonder, is this learning like as in consciously learning something, like as in hopefully how you're learning right now about some different parts of tolerance, or how you might learn other subjects in your studies? Not exactly. This is learning in a much more basic sense. Again, as a bit of a repeat from earlier lectures, this is learning in the sense of classical conditioning, where one stimulus gets associated through to another through repeated pairing. So again, this is an image I've used before, and I apologize for the repeat, but I'm just trying to hopefully solidify this idea that if you pair two stimuli together, like in Pavlov's classic experiments with his dogs, if you pair food and a sound, a whistle together again and again, over time that whistle is enough to elicit some of the effects associated, to elicit some of the counteracting effects that the body normally engages in to offset the drug. So drug compensatory learning is what we sometimes call it. We think of coffee as a collection of cues that can be learned, the sight of coffee, the smell of coffee, the taste of coffee. The body learns or makes the association between these cues and increased heart rate. Whether you consciously realize it or not, your body recognizes that when the smell of coffee is present, when the taste of coffee is present, then the heart rate is fairly soon after will begin to accelerate, because caffeine will be present and interacting with the nervous system. The body makes these associations and begins to decrease heart rate activity even before the caffeine is present, using those cues as kind of early warning to get ready to counteract the effects of the drug. So it's learning in that fairly basic sense. And if you want to build up tolerance to a drug very quickly, you could even take advantage of this by using the same drug in the same way each time to maximize the chances for classical conditioning, maximize the chances for behavioral tolerance. So if you wanted to become particularly tolerant to a drug, I can't say I'd really advise this, but let's say you took a relatively benign drug like caffeine and thought, I want to become tolerant to caffeine. I want to get to the point where caffeine has little or no effect on me. Well, one way to do that would be just to drink the same dose or take the same dose of caffeine, the same size cup of coffee, the same brew of coffee at the same time of day in the same place repeatedly over time, so as to really maximize the chance for your mind to learn all the cues associated with that drug and be best able to mount its counteracting or opponent process. What if you can't do this? Can changes in environmental cues cause changes in tolerance? The answer to this actually is yes. If you use drugs in a new environment, or you use drugs in a different way than you've used them before, you can actually decrease your behavioral tolerance to the drug and can increase the risk of an overdose. I didn't include a slide on this in this particular lecture because this lecture is already getting a bit long, as I'm sure you've noticed, but there are some examples in recent years of celebrities who have died from drug overdoses. One of them was the actor who's in that TV series Glee. I think his name was Corey Monteith, young guy in his 20s or early 30s who died from a heroin overdose. He'd previously, I'll have to look to see if that's heroin. I'm pretty sure it was a heroin overdose. I'll link to a news story on Blackboard to be sure I get my facts right. But essentially, he was someone who had had problems with drugs previously in his life, had undergone some treatment for drug dependence, and had at least allegedly been clean from using drugs for a period of time, and then went on to use drugs in a kind of a new environment. He went, used drugs in a new city, a place he hadn't used them before, and he died from an overdose. It's possible that what happened for him is his body didn't have those warning signals anymore. He'd lost some of his behavioral tolerance for the drug, and the body was less able to counteract the effects of the drug. And thus he unfortunately had a toxic reaction. He overdosed and died. There's a really classic series of studies that have been done on this phenomenon with non-human animals, with rats, where Rats can be given across different days increasing doses of heroin, or really almost any drug, but let's say heroin as an example, such that over time their tolerance increases, and a dose which would initially have been toxic or even fatal to the rat is no longer fatal. Sort of over time, give them the same dose of heroin, allow them to get tolerant to it, then slightly increase the dose, allow them to get tolerant, slightly increase the dose, allow them to get tolerant. And by the end of this cycle of dosing, you have the rat taking a dose of heroin which would have previously killed it dead. If you take that same rat and move it to a new cage, not even necessarily a cage which to a human experimenter seems particularly different, and then give the rat the same high dose of heroin, it will die. The reason it will die is it doesn't have the cues of its environment that were associated with that drug, with that dose of heroin, to help the body get ready to counteract the effect. It doesn't have those early warning signals to mount its opponent processes. So again, this phenomenon has been observed very reliably in non-human animals, where we can do experimental dosing very easily and safely for humans at least. We see things like this in human users who use drugs in a particular environment and then move to a different environment or use them in a new environment and experience decreased tolerance, decreased behavioral tolerance. Okay, so the important idea here is that tolerance is a complicated phenomenon, especially behavioral tolerance, and the setting that drugs are used in plays an important role for kind of laying the groundwork for that type of tolerance. But all forms of tolerance that I've talked about have to do with this basic idea that with repeated use, the effect of a drug decreases. And there are some attempts to explain why this happens, to explain why the different types of tolerance work the way they do. Okay, preview of what we'll cover next time. In our next lecture, we'll just be covering, we'll get into talking about specific drugs in particular, and I'm going to focus on caffeine in my next lecture because I've used so many slides about coffee in this lecture. I'm certainly feeling the urge to use some caffeine and then actually to talk about caffeine. Maybe you are too. If you have a chance, just step back from the computer, step back from the tablet, maybe make yourself a cup of coffee or a cup of tea and relax. And when you do, I'll be here with the next lecture for you to check out. So thanks so much for your attention, as I always say, and I'll see you in the next lecture. Bye-bye.